Hey Bio, how you doing? Welcome back to distance learning session number 5 of biology. Today's date is April 29th, 2020, and we are going over part 2 of your body's third line of defense, which is entitled the HMIR, the Humoral Mediated Immune Response. This part 2 is much more specific, and it is very effective in terms of conquering pathogens that have invaded your body, that are trying to set up shop and form disease. So before we get into the nuts and bolts of part 2, I kind of want to take you back to where we were last week when we were going over part 1, the CMIR, and we had this little scenario that I was kind of going down the road with you guys as we went through this. So let me take you back here, and hope you can see that okay. Said that this was you. And the scenario was that on a Saturday, your mom tells you, hey, we're going to go down to Yuba City and you're going to hang out at the mall all day. So you got nothing better to do up here, so you drive with mom all the way down there. She drops you off. And as you're hanging out, you run into the one hot person that is found in Yuba City. And you strike a little connection, you spend the day with them. And as the day ends, when mom texts you to come back to the car in the parking lot, that one good-looking person gives you a sweet goodbye kiss. But unfortunately, they give you a little bit more than just a goodbye kiss. They give you the Yuba City red macaroni virus. And as you're riding home with mom, and mom's asking you about your day's events, that Yuba City red macaroni virus is going into your body. And it's doing what most viruses do. It spreads. It starts infecting your cells and starts to spread throughout your body. Now, you don't notice very much other than the fact that you got a little tickle in the back of your throat, starts a little scratchy, gives you a little cough. But by the time you get home, you're feeling a little cold and clammy, your temperature's rising a little bit, and you just don't feel 100% because that Yuba City red macaroni virus has infected you. And if you let it go, it is going to set up in terms of disease. So, as we rolled through the steps of the cell-mediated immune response, step 1, I believe, was when this guy shows up. This was Mac macrophage. Mac macrophage turns a corner and sees the pathogen, knows that it doesn't belong, and immediately it engulfs or swallows that pathogen. The second thing that Mac starts to do is, we said, displays the pathogen's antigen. Most pathogens have these pokey things on them that are called antigens. Every pathogen has a different, unique antigen. So Mac, as it swallowed the Yuba City red macaroni virus, is displaying this antigen. Now, Mac needs to call for backup, but as we know, cells can't speak, so Mac secretes a chemical that is called interleukin number 1. And as soon as interleukin number 1 is secreted into the bloodstream, the one cell that responds to it shows up, and it's none other than this guy, our hero, Helper T. Helper T shows up, says, yo, Mac, what do you got? Mac shows the antigen from the Yuba City red macaroni virus. Immediately, Helper T recognizes that antigen, and Helper T calls for backup as well by secreting its own chemical, interleukin number 2. When interleukin number 2 is secreted, everybody shows up. You got the B cell. You've got the memory T cell with a digital camera. You've got the ninja, the cytotoxic killer T cell. And then you got the general, the chicken, the suppressor that kind of hangs out in the background because the suppressor knows that there's active pathogens on site. So at this point, everybody shows up, and Helper T tells killer T cell to start going in and killing all of your infected cells with this stabby probe because, as we all know, killer T, cytotoxic T, kills by the process of lysis. Lysis, as you might remember, is death by stabbing. So killer T stabs all your infected cells. Cytoplasm leaks out, and all of these pathogens, along with your infected cells, die off. Okay? After that, killer T bugs out. Memory T shows up, videotapes all the pathogen bodies around so that if you ever get reinfected by the Yuba City red macaroni virus, we're going to know how to fight it. And then last but not least, after everything is secured, Suppressor T comes in and says, guys, you did a great job. We've conquered the Yuba City red macaroni virus. I'm shutting down the third line of defense. You can all go home, and suppressor shuts it down. Everybody leaves. The site is cleared, but there's one problem, and that problem is this guy right here. Okay? There's one Yuba City red macaroni virus that is unaccounted for, and that virus, as time progresses, goes on to infect other cells. And as the virus infects other cells, that virus spreads, because we all know that when viruses attack human cells, they set up those cells as an assembly line to produce more of that pathogen. So at the end of last week, we weren't out of the woods. We were still sick, which brings us to part 2, the humoral immune response. Now, before I get into the steps, which of these guys did not have an active role in part 1, the CMIR? If you said the B cell, you're exactly right. So part 2 of this third line of defense is all about the B cell today, which we will get to momentarily. But now, before we get into this, let me show you how your book wants me to teach this. Your book wants me to teach this with just an overhead here. You got MAC macrophage, helper T, interleukin 2, not a whole lot to this. And the one thing we see here that we've never seen before are all these Y-shaped bullets that are being shot out at the virus or the pathogen. Those guys are called antibodies. But I kind of want to get into this a little bit more my style, a little bit more entertaining if you don't mind. So here we go. Your body's humoral immune response system, we're going to pick up at step 7 right where we left off last week. Step 7, interleukin number 2 and MAC macrophage with antigen stimulate B cells to divide. So what interleukin 2 now is going to do at the direction of the helper T is kick in the B cells. That white B that was on the board a moment ago is going to start dividing to produce specific types of B cells. Okay? These B cell lymphocytes are going to do the following. They're going to make these Y-shaped bullets, as I like to call them, that are called antibodies. And these antibodies are going to be able to grab a hold of the pokey antigens on the pathogen. And when that happens, when they bind, it's going to inactivate them. In other words, it's going to stop the virus from working, reproducing, and ultimately it's going to kill the virus. Okay? The way they do this, we're going to take a close look at this, is the B cells are going to look at the specific shape of those antigens that are poking out. Remember, as I said before, most pathogens have different shaped antigens, and it's the shape of the antigens that is the key in killing the pathogen by making specific types of antibodies. Okay? Here's a closer look at a different type of virus, and as you can tell, it's got multiple different shaped antigens. And it's the shape of these antigens that we need to look at to kill these things, so to speak. Here's a great example. Here is a virus, and here is the pokey antigen that is sticking out. You guys can see it's got this funky shape over here. B cells are going to produce those Y-shaped antibodies that have the exact same shape as the antigen on the pathogen. So when these antibodies lock hold of the antigen, it's going to stop the pathogen, the virus, from working and essentially going to kill it. Okay? Now, here's the problem that we face. Whenever we get infected by some new type of pathogen, they've got brand new shaped antigens. And we have antibodies from other sicknesses that we've conquered, but these antibodies just won't work because they won't grab a hold. So every time we get infected with something new and it's got different shaped antigens, our bodies have to go through this specific process to make new antibodies that can latch onto these. Very specific. Here's a great example. You guys have this in the slideshow that I gave you. Here are 1, 2, 3 different shaped antibodies that have been produced to kill different pathogens. The one on the far right has circular shapes, and this antibody would only be effective in killing this virus that looks like a jack, because this is the only body that can latch a hold on it. Okay? This antibody right here, this bullet protein with the triangular shapes at the very ends, would only be effective in killing this virus right here that has the inverted triangles. The antibody on the far left, which has square-shaped notches on them, would only be effective in killing this virus right here that has the square-shaped antigens. So as you can see, different pathogens, like these 3 viruses, depending on the shape of their antigens, our bodies have to make very specific antibodies to conquer those pathogens. Now, the last time we actually had a pandemic was in 1918, and that was known as the Spanish American flu. This flu killed more people worldwide than all people that died in all wars combined. A massive influenza. And again, the last time we had a pandemic in the United States or the world, which was 102 years ago. The influenza of 1918 was an avian flu. It was a bird flu. And what was unique about this avian bird flu is this bird flu was able to have what we call viral sex with a human flu strain. They swapped genes. And when they swap genes, they're able to produce antigens unlike the human body has ever seen before. And then, once they produced this super virus, this super flu that was part bird, part avian, and part human, then this magical thing called zoonosis began. Zoonosis is when a virus is able to jump from one species to another. So after they swapped genes and made very specific antigens that poked out, this avian flu was able to jump by the process of zoonosis from birds to humans, which is why it killed so many millions of people back in 1918, because our bodies had never seen antigens like this before. Speed up to 1997, fast forward so to speak, we had another round of what was called Asian bird flu. During this time period, this was another avian flu that was able to mutate, so to speak, with a human strain, and it produced kind of a super bird flu. But this bird flu really didn't kill a whole lot of people in 1997 because it was isolated in certain parts of the world. Culturally speaking, when 1997 the Asian bird flu hit, it was mainly in different countries throughout Asia. And what the World Health Organization found was that young boys in Asian countries were dying more than anyone else. And the reason why was more cultural than anything else. This avian flu had jumped from birds to humans, but the reason why young boys throughout countries in Asia were dying is because culturally it was their job to kill and feather and clean chickens, of all things, so that their family could eat chicken. So they were in direct contact with that avian flu. Once the World Health Organization realized this, they basically killed all the livestock throughout Asia, which decimated that avian influenza, and the population, the human population, was able to rebound. In 2009, we saw another avian flu hit, but it didn't turn out to be avian. It was called the swine flu. In 2009, a huge avian flu came about, and again, it swapped genes. We had viral sex producing a very strong avian flu. But when zoonosis hit, this was the strange thing, we were expecting it to jump from birds to humans, but it didn't work out that way. When zoonosis took place, it jumped from birds to pigs. That virus was able to jump from bird species to pigs, and these pigs were getting sick and dying from it. But then zoonosis took place again, and it caused this swine flu to jump from pigs to humans. So what did we do in 2009? We basically decimated all pig farms and destroyed a lot of pigs throughout the world so that we could curtail this virus from spreading to humans. So here we are, the year is 2020, and we're in the midst of another global pandemic, one that we haven't seen since the likes of 1918, okay? COVID-19 is a coronavirus. Now, coronaviruses in the past have caused diseases like SARS, which is sudden acute respiratory syndrome, or even MERS, which is Middle Eastern respiratory syndrome as well. They have been devastating diseases, but none the likes of COVID-19 that we've seen so far. So where did COVID-19 come from? Where did it originate? Why is it so deadly? Here's what we believe at this point. And keep in mind, this intel that's coming in is fairly fresh. This is kind of cutting edge, so we're not quite sure, but more studies are being done on this. It is believed that the coronavirus originated in bats. And viral sex took place between the bat and this guy, which is a lizard-like creature that's called the pangolin. Okay? So not only did it swap genomes, but through zoonosis, it was able to jump from bats to pangolins. Now, interestingly enough, pangolins are supposedly a delicacy in countries throughout China. So it is believed in some studies that the virus was able to jump by zoonosis from the pangolin to humans. But every time that it jumped, it swapped the viral genes between bats, pangolins, and humans, and created antigens unlike our bodies have never seen before. So when this virus has been infecting humans, these brand new antigens our bodies have basically never seen before. They've seen similar ones with SARS and MERS, but these are so new and the viral reproduction rate is so quick that our bodies are having a hard time catching up with this third line of defense. So this is the information that we know as of now based on COVID-19. So here we are. We're going back to you. As you were infected by the Yuba City red macaroni virus, and now what we're going to do is get this guy into play, which is the B cell. So let's talk specifically about the B cell. Step 8 says this: B cells show up and they start dividing in response to the antigens. And they produce two types of cells. The first type of B cell is called the plasma cells. Plasma cells are, as I like to say, the machine gunners of sorts. Okay? These are B cells that release those magical bullets that are called antibodies at a rate of approximately 30,000 per second. That is the most intense machine gun ever possibly imagined, but they're not bullets, they're actual antibodies that are taking place. So let's go back here. We've got the Yuba City red macaroni virus, and I want to show you something. The B cell is called in. The B cell is going to look carefully at the antigens on the Yuba City red macaroni virus, and the B cell is going to divide, and it is going to produce very specific B cells to that Yuba City red macaroni virus. Here it is, the plasma shooter, the plasma cell. When the plasma cell shows up, it has made these specific antibodies, which are Y-shaped bullets, and it shoots it at a rate of about 30,000 per second. So here we go. It starts unloading these Y-shaped bullets like crazy. 30,000 per second. As each antibody latches on, the antibody's ends are specific to the antigens. So each antibody is going to latch on to the pathogen, and once it latches on, it stops the virus from working, and essentially it kills it. And so I'm just going to dump all these extra antibodies up here because this is one intense machine gun. The plasma B cell fires like crazy. Those antibodies, as I mentioned before, bind to the pathogen's antigens, inactivate and destroy them. Now, one last B cell is going to be produced, and we call this the memory B cell. Memory B cells, get this, are the most important cell that you could have in your bloodstream throughout your lifetime. They remain in the bloodstream for very long periods of time and protect you against reinfection. Okay? I like to say that memory B cells are snipers. Okay? So here's a memory B cell that's specifically made for the Yuba City red macaroni virus. How do you know? Because it says memory on it. Okay? So now, I want you to see this. Let's say the plasma with the machine gun has totally destroyed all these Yuba City red macaroni viruses. Okay? So what's going to happen now is that suppressor is going to come in and say, hey, great job. You guys totally destroyed the Yuba City red macaroni virus. It's done for sure. It's gone. Okay, I'm shutting down the immune response. You guys did a great job. These antibodies are going to remain in your bloodstream for a couple months because they're proteins and they're not going anywhere. Okay? So your body shuts down the immune response. Now, let's say that 6 months has gone by and all these antibodies are now gone, and you're silly enough to go back to Yuba City to see that one hot person, and at the end of the day, they give you a kiss, and they give you that Yuba City red macaroni virus one more time. They've reinfected you, and it goes in and it starts reproducing. Okay? So, if that's the case, your memory B cells, which are the snipers, are going to come in. And those memory B cells are going to recognize the antigens on that pathogen. And they are going to lock and load and shoot these Y-shaped bullets one at a time. Target engaged, fire, boom. And it is going to take out these pathogens one by one to ensure that you're never reinfected and get sick again. When we've built up immunity, it's because we have these memory B cells. There's two ways to get memory B cells in our bodies. Number one, to survive the infection, like I've shown here. The other way to get memory B cells is by a vaccination, which is an injection of weak or dead pathogens so that our bodies can produce these memory B cells. So memory B cells are by far the most important ones of the entire immune response. Okay? Usually last a long time, prevent us from being reinfected, and these memory B cells have the correct antibodies to latch on to those antigens from the pathogen. Okay? Whew! That's a mouthful. Hey, I've said a number of things about the 1918 Spanish-American flu, the last pandemic we had, talked about the 1997 bird flu, talked about the 2009 swine flu, talked a little bit about the pandemic that we're undergoing right now. If you guys have any questions whatsoever, comments, concerns, this is interesting stuff. We're all living through this. Drop me an email. Don't hesitate. Until next week, hope you guys have learned a lot. It's kind of interesting stuff. Hope my toys were good. And you guys stay classy. Take care. Hey, I totally forgot one more thing. So you have these memory B cells against the Yuba City red macaroni virus. You know you're safe. But let's say this. Let's say you're bored and mom drives you not to Yuba City, but to Marysville. And let's say the same scenario takes place. You meet up with the one hot person in Marysville, and when you leave, that person gives you a kiss, only they give you the Marysville blue bonnet virus. And same scenario applies. You guys go home, you're starting to feel sick. So here's my question to you guys. Will the antibodies from the Yuba City red macaroni virus be able to attach and kill the Marysville bluebonnet virus? Yes, no? The answer is no, because these antibodies do not have the same shapes to latch on to the Marysville bluebonnet virus antigens. So your body's going to have to go through this whole immune response so that your body's B cells show up and make not red B cells to kill this, but other specific B cells. So try to imagine how many different types of B cells you have flowing through your bloodstream for different types of colds, flus. If you've ever had a vaccination for mumps, measles, rubella, for a disease called diphtheria, for pertussis or tetanus, if you've ever had shots, all those different diseases that I rattled off that you've either had or you've had vaccinations for, you have all these different types of B cells. Stay classy in you.